Get everybody into the room. Good evening, everybody. My name is Ann Thompson. I'm the Assistant Director for Public Services here at the Essex Library, and it is my pleasure to welcome you tonight on a beautiful evening to have a talk with Dr. Paulo Verardi. And we're going to obviously talk about COVID, the vaccines, its variants, and he's going to give us the wealth of his knowledge in 60 minutes or less, and we're going to open up to questions. Just a few housekeeping notes as we open the meeting. I have a poll here. The State Library asks me to ask you how many people are viewing your screen. So if you could let me know very quickly how many are sharing your screen tonight, and then we'll get that out of the way. While you're answering that, I'll just let you know that we can take your questions this evening. It's going to be a Q&A after a quick presentation. And if you want to put your questions into the chat, or Dr. Verardi has said that you're also welcome to interrupt him. If a question occurs to you and you want to unmute your microphone and shout one out, that would be great too. I do have a couple of questions that got emailed earlier, so I will offer those up. Thank you for answering the poll. I'm going to go put that away in 3, 2, 1. And we'll get that off the screen. And with that, Dr. Paulo Verardi, thank you very much for joining us tonight. He is an associate professor of virology and vaccinology in the Department of Pathobiology at the University of Connecticut up in Storrs. He's an investigator at the Center of Excellence for Vaccine Research at UConn. He received a Bachelor's of Science in Biology from the Federal University of Rio Grande do Sul in Brazil, and earned a PhD in Comparative Pathology at the University of California, Davis. He joined UConn as an assistant professor in 2008. Dr. Verardi has a broad background in virology and immunology with a particular interest in vaccine development, which is obviously of pointed interest to us tonight. He has worked on vaccines for a number of human and animal viruses, including smallpox, AIDS, rinderpest, Rift Valley fever, the Zika virus, and of course, SARS-CoV-2. I think that I said that correctly. I hope so. Anyway, with that, here is Dr. Verardi.
All right, I hope that everybody can hear me well. Thank you very much, Ann, for the invitation to do this. It's really a pleasure for me to do so. I hope that I can answer some of the questions that you all may have for me today. And like Ann said, feel free to ask me any questions on any time. So I'll give a very short presentation now. And in fact, let me get it started before it gets too late here. So, and let me see. So, Ann, just tell me if you see the full screen again.
Yes, yes, I can.
Okay, perfect. So, I'll give a short presentation just for us to have something to look at, and perhaps to make you think of some questions for us to discuss later on. Okay, and like I said, I'll be more than happy to answer any questions, and even during the presentation you can stop me, but I'll try to leave enough time for us to discuss whatever thoughts or questions you may have later on. So the important part here is really it's not for me to lecture you today, but really I'll give an opportunity for you to ask questions. So, and I'll just have to remove something from here. Well, I'll do it later on. Anyway, so what a 15 months it has been, right? So, you guys may not realize this, but it was on March 13th. It was a Friday that some of us left our offices from work and never came back. In fact, my wife left she also works here at UConn. She's a staff member, and she never came back up to this day. She left on March 13th for her last day, in her office. And now we are, June 15th, so 15 months later. Actually, I just copied this from the City Post, back on March 14th, which was a Saturday. The news really happening hour by hour, right? And in fact, at 11 a.m. that Saturday, the first casualty happened in the Northeast here in New York. An 82-year-old woman, died that Saturday. I remember that Saturday very well because I was very busy helping to write an article for USA Today because at that point, the pandemic was kind of happening, but we're kind of all lost, and people didn't even know what a virus was. So I was working with this reporter, Joe, and really trying to answer some basic questions. What is a virus? What, are viruses alive? How do you kill a virus? And why does a virus make us sick? Can vaccines or medicines help fight a viral disease? That type of thing. And it's interesting because I was thinking about, 15 months ago, I actually went back and read this article again, just to make sure, what type of predictions maybe I made or whatever, I said at the time, was it right or wrong? And luckily everything, was kind of right. So I'm glad that I did that. But I'll tell you very briefly, what is a virus? So a virus actually, it's something very simple. It's a particle, and we call it, if you're referring to the particle, we call that a virion. So virus is just a general name, but a virion is for the actual particle. And it has a nucleic acid, it has a piece of DNA or RNA as part of its genome, and is encapsulated, is surrounded by protein. And in fact, the simplest, one of the simplest, not the simplest, but one of the simplest viruses is a plant virus called tobacco mosaic virus. And it was actually the first virus to be discovered about 130 years ago. And that's all it is a piece of RNA that is encapsulated, with this cold protein. So there's this protein here that just, wraps around it, and it forms these virus particles here, the virions. And in this case here, they are helical, they form these tubes. And the tobacco mosaic virus, the name mosaic, comes from the fact that it creates these mosaic patterns in the leaves of infected tobacco plants. So that's a little bit of history for you, probably more than you needed. But viruses do need cells to replicate. They are not capable of reproducing or replicating by themselves. So they need cells from a host to do that. They are really intracellular parasites. They need to enter the cells. When they do that, they hijack those cells. And what you see here is a number of different cells in cell culture. And right in the middle, you see there a little hole in that monolayer. And in fact, because they cannot reproduce or replicate by themselves, they are technically not live. They're just a piece of DNA and protein. So we don't really consider them live, although this is a philosophical type of question. But here you see, obviously this is a sped up video. You see that a virus infected some of those cells in the center, and it's kind of spreading out. And what you see is not the virus spreading out, but what you see is just the cells. They kind of round up. They become rounder when the virus infects them. So that's why you see this ring that's forming. And in fact, if you take the cells and you stain them, if you just really stain with a dye, you'll see that there are holes in the monolayer. This is the regions where the cells died. So this whole thing here is that plaque. We call it a plaque that has formed. For this particular virus here, it forms about 2 days later on, okay? So you see that they need cells to replicate. They technically are not alive. They hijack the cells, and they use those cells for their own purposes, which in this case is to make more copies of themselves. And again, what a time to be a virologist and a vaccinologist, because I am both a virologist, I study viruses, and I also work in vaccine development. So I am a vaccinologist. And, back a year, 2 years ago, I had to explain what vaccinology was. And vaccinologist, the autocorrecting word doesn't still recognize it, so it's not a common word. But because of that, I have been asked to do lots of things in this past 15 months, both nationally, in this case, an article talking about variants, which we'll talk more about later, even given, interviews for a worldwide audience. And 15 months ago, if you asked me that I would be doing this type of things, I would say, no way, but that's how much we are in demand these days. But let's get serious here. Let's see what the situation is today in the US. So this is straight from the Johns Hopkins dashboard, the COVID-19 dashboard. We are doing pretty well. I mean, you can see here that the number of infections are going down, the number of deaths are going down. Worldwide, the situation is a little bit different. And let me just, sorry, I'll have to remove something from here, because otherwise I can see the presentation myself. So we are back there now. So the situation worldwide is a little bit different. We are doing much better now here in the US than worldwide, but things are kind of calming down. It really depends on the country, right? Still number of deaths, per day, very, very high. We are having an average of about 10,000 per day still worldwide. Much, much lower, level here in the US. Things are getting under control. But, today, I believe we reached the 600,000, number of deaths. So 33 million, infected. And this is the official number. It could be higher than that, 600,000, deaths caused by COVID-19 just here in the US, about 3.8 million people worldwide. So really significant, loss of life just because of this virus. So we'll get back to this a little bit later on. I want to also talk to you very briefly about vaccines. I normally do, this very slowly. I don't want to spend too much time doing this today. I'm just going to very briefly talk about the types of vaccines, and then I'll tell you a little bit about their safety, efficacy, and how fast you can develop them, that type of thing. But there are a number of vaccines that are developed in a more classical way, things like the measles vaccine, or maybe the Salk polio vaccine, which is an inactivated type of virus. So it's this will be the pathogenic virus that would cause disease. This virus here has been killed, usually by chemical means or some other ways. So it doesn't cause disease, it doesn't replicate, but it keeps, it has a structure that's obviously, resembles the pathogenic virus. It is derived from the pathogenic virus. So you end up developing an immune response to it. More recently, most of our vaccines actually fall into this category here. They are recombinant or derived by recombinant DNA technology. Most of them are protein subunit vaccines, like the hepatitis B, the HPV vaccine. And some of them are, with this more modern, even more modern technologies. Protein subunit vaccines are very common now. Viral vector vaccines are less common. In fact, the adenovirus that's used for the Johnson & Johnson vaccine, for the AstraZeneca vaccine, is sort of one of the first ones to be approved, for human use. It's a non-replicating one, so it's safer. And then, we have the DNA and the messenger RNA vaccines. And again, this is the newest type of technology that we have. And in fact, those were the first ones approved for emergency use in the US. But you have to think about safety and efficacy. You have to ponder, a number of different things when you are developing vaccines. And so you have many pathways to take, you have many questions, to answer, that type of thing. But we have done, and I say we, we as a human species, as the human population, we have done a great job. The WHO actually keeps track of the vaccines under development. And there are currently 102, so more than 100 vaccines in clinical trials. This is in clinical human evaluation throughout the world. And you may say, well, this is, why do we need so many, that type of thing? Well, remember, we do have a shortage of vaccines, not here in the US anymore, but worldwide. So we need as many vaccines as possible. And I'll explain to you in a little bit. We have 3 that are approved for emergency use here in the US, the Moderna, the Johnson & Johnson, and the Pfizer. 2 of them are messenger RNA vaccines. 1 is an adenovirus, non-replicating vector. Throughout the world, we have a number of other ones that have been approved, including some that are inactivated viruses, some that are also viral replicating viruses, and some other ones. So we have a number of them that have already either being approved or under, advanced clinical development. But each one of them, again, offer advantages and disadvantages. And in particular, you have to think about cost, because in poor countries, cost is significant. Is a significant barrier for vaccinating the population. Thermostability, because not everybody can have those expensive freezers to keep, for example, the Pfizer vaccine, right? So thermostability, transport of the vaccines, all those things becomes important. And also how easy it is to manufacture them. Again, if you need really very expensive, complicated manufacturing facilities that are not available, in countries that are straining their resources, that may not work. What worked for us here in the US may not end up working, for countries in Africa, for example, that type of thing. So it's good that we have, a variety of different vaccines. And in fact, just in my family, I'm originally from Brazil. Just in my family, I have my immediate family, and this essentially is, myself, my kids, and my sisters, in this case, my mom. We all have received, each one a different type of vaccine, essentially. Anything between the Moderna, the Pfizer, the AstraZeneca, one called CoronaVac, an inactivated one that's given in Brazil. And I think there's one more, in the Johnson & Johnson. So, we just, because we live in two different countries, we have received already a number of different types of vaccines. And that's important because my mom was able to get vaccinated with the CoronaVac vaccine early on because it was very easy to manufacture, for example. It was manufactured in China. So some of my, one of my sisters, was able to get the Pfizer vaccine more recently, that type of thing, or the AstraZeneca vaccine. So, all right, and why, vaccines, work? You have really a number of different types of vaccines, and I just showed you, Johnson & Johnson, Moderna, Pfizer, Novavax is another one that's coming up, right? It will likely receive the approval, soon enough. Or you can have actually the pathogenic virus. What they do, they get recognized by our immune system. Our immune system has an innate way of sensing virus particles or components of the virus, or things that are added to these vaccines to activate the innate immune system. And when that happens, specific T cells are activated. CD4 T cells, they're called T helper cells, and CD8 T cells, they're called cytotoxic T lymphocytes. So these are very viral specific T cell responses. They recognize particular amino acid sequences, essentially, from the virus. And these helper T cells also help, B cells to get activated and to produce viral specific antibodies. So it's really these T cells and these antibodies here that offer us protection. And I'll show you how in a little bit. Most people know about the antibodies. Most people kind of know about the T cells, but don't know how they work. The antibodies, they work mainly by one method, and it is by neutralizing the virus. So the virus particle needs to bind to cells to be able to enter the cells. Cells just, will not take these viruses if they do not bind to them, and they need to bind to a specific receptor on the cell. So they actually have to recognize a component on the cell membrane to be able to bind and then enter the cell and then, start the replication process. And what happens is that antibodies, if they bind in a very specific way here, they may actually block the binding of the virus particle to those receptors on the cell, and therefore prevent infection. Either because they prevent the binding, sometimes they can also prevent this fusion process that occurs. So when they do that, these are called the neutralizing antibodies. They block the virus, they prevent infection in the first place. So if you have a lot of these neutralizing antibodies, you are very likely to not even get infected. You maybe get exposed to a virus, but not get infected. The other way by which our immune system protects us from viruses is those T cells. And I'll show you an example here, one type. So when the viruses, when they infect the cells, they produce a lot of what we call progeny virus, right? They produce a lot of copies of themselves, and they get out of the cell, in a number of different ways. They can butt out from the cell, for example, and they can infect new cells. So an infected cell becomes a viral factory. It's unknown to the cell because the virus is infecting it. There's nothing that the cell can do. I mean, the cell usually actually does try to stop that, but is usually unable to do so. And here you see a lot of viruses actually budding from, these are influenza viruses budding from the cell membrane. Well, those cytotoxic T cells, what they do, they can actually recognize that a cell is infected in a very specific way. It's a complicated way, but it's a very specific way. And then they can actually kill those cells. And it sounds like counterintuitive. Why would some of our cells try to kill our own cells? Well, they're only going to kill the virus-infected cells. And if they do that before virus replication is completed and before these viruses bud out from the cell, so to speak, then they are going to have a big effect because you're essentially killing their little factory, right? You are stopping the factory producing more virus particles. So these cells are called cytotoxic T lymphocytes, or CTLs, and they really help to clear the infection in the host. So it's really this action of the antibodies preventing infection, these cytotoxic T cells clearing the infection. These two work together, and they eventually either prevent infection in the first place, or if you do get infected, they help to minimize infection and eventually clear that infection. So this is what our immune system does, okay? And we usually talk more about antibodies. We don't talk much about these T cells that are very, very important for viral infections. And we were able to develop vaccines really in record time by this process called gene to vaccine, because in late 2019, we had those cases of pneumonia in Wuhan, China. By January 10th, the SARS coronavirus 2 genetic sequence, it was called then the novel coronavirus, was actually, the RNA was actually sequenced, and the sequence was released to the world, okay? People here in the US, people in Europe, people everywhere. And this is just a picture of that, genetic sequence, in fact. So one of the things that we can do, we can take this digital information here, just, the composition of the RNA, or and we translate that into, whatever protein it produces. And we can actually synthesize our own version of that, and usually in the form of a DNA sequence. And with that DNA sequence, we can produce any vaccine types, any of those recombinant vaccine types that I told you before, protein subunits, viral vector ones, or some of these DNA and messenger RNA vaccines. So we actually don't need to have a viral sample. So by January 10th, people here in the US and in Europe were already working on a vaccine, even though they, will not see the actual real virus for a long, long time. So because of that, again, we can start vaccine design as early, as soon as we have those genetic sequences, and actually vaccine development truly, honestly, started, on that day already in the world, because the big vaccine companies, understood early on that this could become, a major problem. So they decided to get started with it right away. And, the SARS coronavirus, I guess you guys all, know how it looks like. The name corona comes from the fact that it has this crown appearance under electron microscopy. So it looks like a crown. And that's what the name, it comes, corona, crown. So this is actually an original SARS coronavirus. It's not a SARS coronavirus 2, but they really, under electron microscopy, look about the same. But they do have this spike protein that most of us know what it is now. Is responsible for this crown. Is the one that actually binds to the cell receptor. Oh, I'm sorry. That's called ACE2. So ACE2 is this receptor on cells, and the spike protein is the one that binds to it and allows the virus to enter that cell. So neutralizing antibodies are made against the spike protein, and therefore the spike protein is essentially the antigen, the portion of the virus that's using all of these vaccine types that you are familiar with: AstraZeneca, Johnson & Johnson, the Pfizer, Moderna, and so on. And but there were some challenges. Normally, it takes in the past, maybe 5 years, the long past. In more modern times, 10, 20 years to develop a vaccine and actually get it licensed, approved. We were in no condition to wait 5 to 20 years, right? So the good news is that we had a lot of experience with SARS coronavirus, the original coronavirus from 2002, 2003, and also with another kind of similar one called MERS coronavirus that showed up in 2011, 12. So we had some experience with it. And because of that, we went straight from the lab, essentially, almost immediately into human clinical trials. And normally we go into preclinical testing, we have to testing animals, that type of thing, but we were able to do so. We actually went back to animal testing as well to make sure, the things were correct. And because of that, we're able to get emergency use authorization of these vaccines within a 1-year time frame. In fact, slightly less than 1 year, which is really extraordinary, but necessary for a pandemic type of situation, right? Now, there's always breaking news. Today's breaking news is that the CDC now classifies, has a new variant of concern. And if you're wondering, it is the new Delta variant. So the Delta variant was already considered a variant of concern by the WHO because of the situation in India, and not here in the US because it was relatively uncommon. But now here it's becoming the fastest growing, variant in the US. So the US has declared, a variant of concern as of today. There are some studies showing that indeed it spreads even more easily than some of the other variants, like the Alpha or the UK variant, and that it may, again, even cause not necessarily more severe disease, but there might be an increase in hospitalization. Some of the data, is showing that. So something to pay attention, because we seem to be doing pretty well right now. But we can't really, just ignore the fact that there are new variants in. So you're going to have to learn, remember your Greek alphabet, because of all of these new variants here. So, we have the alpha originally, and now it's a much easier than saying B117, right? So the original one from the UK, the beta one from South Africa, the gamma, the P1 from Brazil, the delta, that's the one from India. And then also we have a couple of US strains, and those are all considered, so these are from California. They are, the WHO calls them the Epsilon, and they are all from, they are here from California. So they are considered by the CDC variants of concern. I believe the WHO does not consider them yet variants of concern because, again, they are not distributed worldwide. But and you keep going. Okay, it was like hurricane season last year. Remember that we started getting into the Greek alphabet and kept going, and we're gonna keep going. Okay, they have this way difficult to understand nomenclatures here. That type of thing. If you guys want to know more about that, I can talk about this as well. But it's been 20 minutes, 25 minutes or so. I've talked a lot, so now is your time to talk, ask questions, that type of thing. So let me stop my presentation here. You're welcome to send those questions by chat, or better, just unmute yourself, maybe even show your video and ask the questions. I'm here to answer anything that you may want to know that I may be able to answer.
Paulo, thank you so much for that overview of what is happening. It's so much information to even go through each of those slides in great depth would take more than an evening. So I appreciate
that's the problem with it.
Yeah, exactly. I appreciate your ability to narrow it down into the gist of things very quickly and articulately. I think if everybody's been watching the news, then the Delta variant has been the stealing the show, so to speak. And so my first question, and I get to ask first, is why are states, or why is the thinking that if the Delta variant is already around and fairly prevalent around the world, are we looking for a larger outbreak at the end of the summer rather than, at the end of June, so to speak? If it spreads so quickly, why would it take until the end of the summer to create?
In the US, right?
Yes.
Yeah. So one of the things that I forgot, because I tried to do this very quickly, was, and in fact, I may try to show, because this is related to your question. So let me go back to my presentation. Sure. I usually don't like to do, but wrong slide, so I don't want to take too much time here. I just want to show you the graphs already. Here we go. But I'm not, am I sharing?
No, I'm not seeing it.
Yeah, so if I don't share, you guys can see. Here we go.
There you go.
Yeah, so one of the things that I just want to mention is this is the number of people that have been, it's the number of total vaccine doses given. And you see here in the US, we already gave 300 million, more than 300 million doses. Obviously, some of the vaccines require 2 doses. So the actual number of vaccinated people is not, 300 million. But we have already, about 50% or so of the population that have received at least, 1 dose of the vaccine. And in certain places like here in New England, we already, are way, beyond that. And we are probably going to get to a point very soon where we're going to have about 70, perhaps 75% of the population fully vaccinated. So that, may allow us to start breaking some of those, making it, making it a little bit more difficult for this Delta variant to get transmitted from person to person. Because remember, each variant has a certain ability, capability for transmission, right? And but that assumes, a naive population. If you use masks, you reduce the capability for that to happen. If you are vaccinated now, even if it's not everybody, let's say 75%, you're going to reduce, that transmission rate even more. So the effective transmission rate of the Delta variant here in the US is not going to be the same as it was in India, where most of the population was naive, was not vaccinated, and not received the vaccine, right? So you don't expect to see that. So what you expect to see is a small increase in the proportion of the people that will actually be getting the Delta variant. And then it could be that once we, winter comes back, so late summer to, early fall, particularly here, perhaps in New England, but again, we are more protected because we are more vaccinated here. Once you start retreating more inside, that type of thing, a lot of these respiratory diseases, they are seasonal, right? They tend to happen more in the fall and in the winter. So that's when, we may end up seeing more cases here in the Northeast, here in New England. The situation, in the southern states may be a little bit different because then there, a lot less people are going to be vaccinated, and therefore, it will be a little bit easier. Probably those, probably there might be surges in some of those states they're not as vaccinated as we are here in the Northeast. Having said that, the situation in other countries in the world will really depend on those countries, right? In the number of people that are vaccinated. So we really have to look at this in a very regional, local way. We can really, make big predictions about the US as a whole, particularly now that we have, this big variation, this big difference in vaccination rates between different states and so on. So the bottom line is, try to convince as many people to be vaccinated, because that not only helps those people, it helps all of us by this herd immunity effect. So we're gonna see what's gonna happen there. Let me stop this because then I can see the chat.
Okay, thank you. So I have a couple of questions that were emailed earlier before the program. Tom asked, anecdotal evidence suggests Moderna vaccine has more side effects than the Pfizer vaccine. Is there a possible reason for this difference?
There might, and it's funny because some people tell me the opposite. They said, that the Pfizer one, gave them a lot of, so you have, people saying one thing or the other. Pfizer causing more severe, complications after the second shot, and so on. I quite honestly, I'm not keeping track of this, and I don't know really how well people are keeping track of this minor side effects, right? Because most of the side effects are minor. I do have people, that are related people to me that, had some severe, side effects. But it's hard to predict. The Moderna has more RNA, for example, than the Pfizer. So that could be a reason. You have more RNA, therefore you have more reaction. But their composition, they are encapsulated in something that we call liposomes or nanoparticles. They are, and their composition is slightly different. And those types of molecules, they help to enhance the immune response. And in fact, when they do that, they cause inflammation. And if they cause inflammation, one of the hallmarks of inflammation is pain, right? That type of thing, redness, edema, So the reality is there is an inverted correlation. The best vaccines are the ones that are more reactogenic, that cause more reactions. They give you the best immune responses. So what people try to do is a balance, right? Is a dose of the vaccine, an adjuvant, or a formulation that, that will make it acceptable by people, and will still provide, the best immune response. Because, if you really want to make sure that you get the best immune response, you're going to get it, in a certain type of formulation that will make, hurt for a couple of weeks, okay? That, I remember, not being able to lift my arm for a couple of days. Oh, that will last, a couple of weeks. So they do that. They try to manage, the reactogenicity of the vaccine, but the problem is the reactogenicity actually positively correlates with the more robust immune responses. So that's all I can tell you. But I really, at this point, from what I have heard, I really don't see much difference between the Moderna and the Pfizer vaccines, to be quite honest.
Okay, I can say from the library point of view, working at the circulation desk, we've heard stories about all of them. So there's
exactly,
really,
yeah.
and
I had,
that's a good thing.
I know. I mean, one of my kids, didn't even say, no, I feel nothing, and then I have a cousin that, really almost, felt like she needed to go get hospitalized. I had another cousin that was, for the second shot, was feverish, and trembling, the whole night, that type of thing. So, and I was perfectly fine. I just couldn't lift my arm, that much. So, and don't pay much attention to that. That's really within the variation that is expected for all of us. And, if you get a big reaction, then you got vaccinated. That's the good news. Okay, you know for sure that you got vaccinated.
Oh, good. Okay. So the second question, Francesca asked, why has the CDC changed the PCR cycles to 28 for vaccinated, yet kept them at 40 for the unvaccinated? Is it more data manipulation?
So Francesca, can you, if you can unmute yourself, can you elaborate that a little bit more? I'm not as familiar with the diagnostic portions of it because I'm not following it very much. I know that the CDC is going up to, for sequencing purposes, if you want to know which strain you got infected with, for sequencing purposes, they go up to, they try to stay below the cycle 28. And there is a reason for that, is because, that will give you, a better, a sequence that's less prone to mistakes, to errors. So it will be a better, reading of the sequence of the virus. I assume, I'll be really honest with you, I assume that for diagnostic purposes, they're not changing the cycle to less than 40. It just doesn't make sense to me, and quite honestly, the operation of it, wouldn't make sense as well. So, Francesca, if you want to say anything, if you want to tell me, where you heard this from, that type of thing, I'll be more than glad to listen to you. But that's all I know, and I'm pretty sure that the so-called CT values or the cycle threshold has not changed for they're not even going to know, quite honestly, the people doing the testing, if you got vaccinated or not. It's all barcoded, that type of thing. So I really doubt that. I would consider that potentially fake news, but I would have to look it up.
Okay. Diane asked, do you expect another big outbreak this winter?
No, not here in the US. So that's the good news. I think we are doing pretty good, particularly here in the Northeast. There is this element of the Delta variant, right? So, we really don't understand the Delta variant too much. If it is really, much more highly transmissible, we could end up even here in New England with some, a surge. It will probably be a small surge because, again, most of us are going to be vaccinated. And you saw, the vaccines actually are working against these variants. They are working in the sense that you either not getting sick, not getting hospitalized, and even if you do get hospitalized, you're not dying from it. That's all good news, okay? But it doesn't mean that you may not get infected if exposed with the Delta variant. So myself, I am vaccinated. I may get exposed to the virus. It may end up actually allowing the virus to replicate locally, primarily in the upper respiratory tract, our nose, nose passages, that type of thing. In other words, it's very unlikely that the virus will be able to travel to my lower respiratory tract, deep into my lungs, and cause disease. But because it's replicating locally here, and that's because the upper respiratory tract is not going to have as much of those antibodies that I told you. And it's also not going to have, those cytotoxic T cells, those type of things. So the virus may replicate here, it may not go anywhere. But during that time, I myself vaccinated, maybe transmitting the virus to someone that's not vaccinated, for example, right? Like, for example, if I happen to have a kid that's younger, than the age allowed for vaccination, younger than 12 right now, or somebody that couldn't have the vaccine, or someone that for whatever reason, chose not to get vaccinated. Or if I travel back to Brazil, in Brazil, it's very difficult to get vaccinated. As I told you, some of my family members were lucky enough. People of my age are still not only, people of my age now in Brazil, only if they have, a particular reason, that would require them to get vaccinated, immunocompromised, that type of thing, they may receive a vaccine. So if I travel to Brazil, I need to visit my mom. It's been 2 years that I don't see my mom. I don't want to, be carrying the vaccinated and protected, but carrying the virus and giving it to somebody else there that has received the vaccine. And and this for me is very close. I have a cousin of mine in Brazil, from Brazil. He's about 9 months younger than me. Not that I'm super young, but he was very strong. He was, so you can see that he passed away. He was very strong. He was an athlete, actually, a personal trainer, and he got COVID in March, and lasted probably 3 weeks and passed away. This was during the P1, surge that we had, in starting February, March. So by the end of March, he passed away. So these things are happening, and I have to be conscious to the fact that even if I'm vaccinated, I still have to try to protect others, right? This is all, it's the right thing to do, in other words, right? So keep that in mind. And because of that, I don't expect us here, going back to the question, I don't expect us here to have an outbreak in the US, in the Northeast, in this winter. We may have, a little surge of cases, that type of thing. I do expect that, that for the following winter, so not this fall, but fall 2023, that we may need to get booster vaccinations. We don't know, for that, winter of fall 2000, fall and winter 2023, 2024. So right now, for I'm sorry, fall 2022.
Got it.
Not 23. I'm 1 year ahead already. I guess I want to get out of this, of this pandemic as quickly as possible. So, so not this fall, in coming winter, perhaps for the other one, we may have to get a booster vaccination. I think it's too early to tell. Some of the vaccine companies like Pfizer, the CEO is saying, oh, we're going to need it. But remember, they are for business. For profit. I want to see the data first, and all of those decisions will be made on data. So we're going to have a pretty good idea, and probably going to have a lot of vaccine stockpile, maybe some updated versions of the vaccine that are going to work even better against some of these new variants. So it could be that in the future we may need a booster shot, just like we do it for flu. Probably not at the frequency of flu, but every once in a while, a new variant may show up that we may need to get a booster vaccination that will be wise for us to get it. So we need to get prepared for that. Absolutely. Okay.
Thank you. I'm very sorry for your loss. It's horrific.
Yeah.
Tom asks, what is your thought about the two hypotheses regarding origin of virus, from wildlife reservoir or man-made in a lab?
Yeah. Yeah, I'll give the answer that almost everybody will give, which is really we don't know, right? There is, we know that coronaviruses. First of all, there are 7 coronaviruses now that infect humans. Until 2000 until the year 2000, there were only 4. And then we had the first original coronavirus, then MERS, and now the second SARS coronavirus. So we have had, prior to this pandemic, 2 introductions of coronaviruses into the human population, and those were by natural means. Okay, we know that now. For the original coronavirus, it took us about 10 years or so, but we kind of figured out what was going on. So it's not so the most logical, the one that most virologists will think as being the most plausible, is a natural exposure type of thing again, where the virus somehow jumped from bats, perhaps to an intermediate host, perhaps straight to humans, and somehow got adapted. Viruses, when they jump from one species to another, they are not very well, adapted to that species. So typically, they will not cause much of a disease. They are not going to replicate very well. They are usually not very well transmissible at all when they change to a new host, like jumping to humans. And then what happens is they have a period of adaptation where they enhance their ability to get transmitted, and so on. And then and that all could have happened back in 2019, somewhere in China. And to the point that it only became obvious, at the end of 2019, when more people and more human-to-human transmission was occurring. So a lot of these respiratory viruses, like influenza and coronaviruses, when they jump into humans, they don't get transmitted very well. We don't have a lot of human-to-human transmission. But eventually, one just finds the right, couple of mutations here and there, and hangs on to the human, get transmitted, and then it adapts, because that's what viruses do. Viruses are just trying, they make millions of copies of themselves, all the time, and they are error prone, so there are mutations, and sometimes some of these new variants that they're made, they just take over, right? They get more easily transmitted, and they just take over. So that is the most plausible, I would say, explanation. Now, the fact that there was a lab in Wuhan working with coronaviruses from bats and everything does raise eyebrows, no question, right? The problem is we have zero access to that, to whatever was happening there, because the Chinese government is not going to let us know exactly what was happening there. So because of that, we may never know what happened. We may try to piece and figure it out, little by little, doing real detective work. We may, end up discovering what happened. But even if that was the case, it was probably just a leak from the lab. It was not something that was engineered to really cause, be really well adapted to humans, that type of thing. It was probably a virus that was somehow, that leaked, and that transmission occurred. But there is no way to know for sure. So, this debate will go on forever. Okay, it's never gonna die. There are gonna be all sorts of, ideas about it, and it's gonna become crazy. There's a lot at stake. I mean, for the country of China, just imagine how much is at stake there, right? So we may not know. And people are saying, well, why were they studying these viruses in the first place, right? Maybe that was a bad idea. Well, you kind of have to know the enemy, right? I mean, you have to know what you're dealing with. So, maybe they were not doing it in the best way. I don't know. I mean, I really have never been to that place. Not a lot of people have been to that place and have access. These are, for biosecurity reasons, they are not places that you can just walk in and pay a visit. So we may not, ever know what happened, but still, I personally think that the most plausible way is a natural way of transmission, because it already happened twice, and quite honestly, this was January 10th, right? January 9th, 10th, that we kind of, figured out what was going on. On January 4th or so, 7th or so, I was giving a talk specifically about coronaviruses, having pandemic potential. There are actually regulations here in the US to work with these types of viruses because they have, pandemic potential, that type of thing. So I was actually giving a lecture abroad, trying to enhance, what we call biosecurity, abroad, in one of the places where murders was discovered. So to avoid this type of thing. But the reality is, people are always going to be, in close proximity to the environment, to wildlife, to animals, is inevitable. So, I would argue that we still need to know and study them, very well. We just have to do a good job, when we're studying, so that we don't create a situation like this. But I believe, that probably, that's what happened, that it was a natural infection, that, and not a lab leak, that type of thing. But who knows? I mean, we're going to have to look for additional, more information, and those are going to be hard to come by, quite honestly.
Sounds like it'll make for a good book, a great science fiction book.
Oh, there will be movies and things, and you name it. Yeah.
For sure. We have more questions. Back to the
side effects. Diane asks, if you only had a minor reaction to the second shot, does that mean you didn't have much of an immune response and your antibodies might be low?
Yeah, so if you did have a good reaction, it's peace of mind because you got vaccinated, right? If you don't have one, the good news is there's no real correlation, okay? So it doesn't mean that if your reaction was, if you didn't have much of a reaction, you're gonna have less antibodies, less of those T cells. That's really the good news. It's just that, when you do have a big reaction, you for sure know that you got the vaccine. And I tell you this because, in certain places in the world, and I'm ashamed to say that in Brazil, my home country is one of them, people were not being given the real vaccine, okay? Yes, so, vaccines are being stolen from places. People were giving, just, water or PBS or something like that, a buffer instead of the vaccine to people. All sorts of things are happening because, remember, everybody's, in a certain place is dying, the hospitals are full. People will do desperate things, right? I heard of people coming with firearms into a vaccination site and stealing the vaccine. So you, and don't think that the US is that different. One day we are going to know who got the vaccine first, and it was probably people with a lot of money, okay, not necessarily. So, it happens everywhere. It's just that in Brazil and other places it's more obvious. So, if you didn't get it, don't panic, okay? Absolutely no correlation there. You just know that it's just that if you get it, you kind of know that you got probably something that had an active component to it. So that's good news.
So back to another one. So what do you say to people who are worried about the side effects of a vaccine? So in a sense of trying to—
Yeah, so there are side effects that are just reactions, right? The arm hurts, that type of thing, or you may have chills, fever, and some of those are uncomfortable. I remember, I myself a couple of years ago, my doctor said, "Oh, you need to have the tetanus shot again." And I said, "Oh, I have a big meeting coming up next week." I said, "Let's do next year," because I said I need to be able to function, to be able to write with my arm, that type of thing. So I said, "Let's do next year." Okay. So even I have, I'm guilty. Okay, I'm guilty as charged. I have delayed my tetanus shot by 1 year because it was too reactogenic. But that's minor. That's nothing. Really, what we are worried about are some of those things like the blood clots, right? Yeah. Things that actually can be really serious. Luckily, those things are very, very uncommon. They are very rare, and they're happening only for certain types of vaccines like that, like the AstraZeneca or the Johnson & Johnson. It doesn't happen to everybody. You have for any type of procedure, any type of inoculation, it's a cost benefit type of analysis, right? And in the middle of a pandemic, I might be more willing to accept certain risks, even if they are not, infinitesimally small. I may still be able to accept that because there's a bigger chance that I may get it, and I may end up getting blood clots from COVID-19, and in fact, that was one of the major problems with my cousin. He had so many blood clots that they were giving him so much heparin that ended up making his situation even worse. So he didn't technically die from coronavirus. He died from all of the things that were happening to him, right? Due to originally to the coronavirus. So including a hospital-acquired infection because of the ventilator, all of those things. So we have to keep thinking about all of those things, right? So that's what I have to tell. The data is out there. Obviously sometimes you do a clinical trial with 40,000 people, of which 20,000 were vaccinated. So if the risk for a particular side effect is 1 in a million, you're not going to see that in 40,000 people, right? You're only going to see that once it goes into the larger population. But again, in a pandemic type of situation, it's a risk that I would be willing to take because I would have a much bigger chance of dying from COVID than dying from a side effect from the vaccine, particularly because a lot of side effects from vaccines are treatable, okay? So we didn't know exactly how to treat the blood clots from the complications from the Johnson & Johnson and AstraZeneca. Now we know a lot how to treat them a lot better because you cannot use heparin. You have to use a different type of anticoagulant. So that type of thing. So we are evolving there. And some people would say, oh, maybe it's a better idea than to just wait, so that, we resolve all of those issues, we figure out what's going on. But then you're taking a risk as well. You're taking a risk because you're waiting. And the longer you wait, the more, the better chances, the more chances you have of actually acquiring COVID-19 and actually having a complication from it or dying from it, that type of thing. So a lot of people, not here in the US, because in the US you have the Pfizer and Moderna, really robust vaccines. But in Brazil, in certain countries in Europe, India and so on, they ask me, well, should I wait because I don't want to take, for example, the CoronaVac that was given in Brazil, that's given in Brazil. That is not as robust. The efficacy in Brazil was shown to be about 50%, not 90%. Although, it's still protected from death. So people were not dying from it. So for me, that sells it to me already. And my advice to my mom, for example, is, because she's 81, she was able to get it a little bit earlier than everybody else, is take whatever you can get first, because what are you going to do? Wait for the perfect one and then die in the meantime? No, don't do that. So it's really a cost benefit analysis. But remember, everything is rare, and I think we have to keep ourselves, keep reminding ourselves of that, right?
Well, you just answered the perfect question, which is, what would you tell your mom to do? And
exactly, and that's what I told my mom. Take whatever comes your way first. Obviously, if it's a real vaccine, not the one that, because there were people, again, selling vaccines. Originally they were not real vaccines. But if it's something approved and so on, go and take it. Don't wait for a better vaccine. And in fact, I'll tell her now, as soon as more people get vaccinated, I'll tell her to probably get a Pfizer or Moderna shot to kind of really boost her immunity, because she got one that was not ideal, was not as robust as the Pfizer. But let other people get theirs first. Okay, let's get everybody out of, let's try to save as many people as possible. So there will be a time that even in Brazil, you'll be able to hopefully, pick and choose, and get a third dose or a booster shot, that type of thing. But we are not there yet. For the rest of the world, we are not there yet.
Right. So this brings up the next question, which is Diane, I believe, Diane is worried that in countries with a low vaccination rate, she's worried that a more virulent variant will develop where the current vaccines are not protective. What would happen then?
That's exactly why we are telling people to get vaccinated, and we need to vaccinate everybody as quickly as possible. Essentially, each one of us is a test tube, okay? And the virus comes and infects us, and we become a test tube for the virus. We become an experiment. And the virus will try, will mutate, because it's an RNA viruses, that's what they do. And they're gonna, and they will try to, especially, and they're gonna try to essentially, get more easily transmitted. By doing so, they may enhance. Usually they don't do that. Usually over time, actually, they become less virulent. Virulent means pathogenic, they cause less severe disease. Although at the very beginning, they may end up, because they're optimizing themselves, they may end up causing more severe disease. So, but they get more transmissible, that type of thing. So the more test tubes we have in the world, the better chances we have to develop new, designer, for lack of a better word, variants that will cause more damage, right? If we can reduce the number of test tubes by getting people vaccinated, so that they will, really limit replication of the virus. And even, some vaccines, like I was just saying before, they may still allow local replication, that type of thing. But that just reduces the chances of us, allowing this virus to diversify even more. Okay, and that's why it's really, a race against time here. We really need to try to get 7.8 billion people in the world vaccinated as quickly as possible. And having said that, the rest of the world is looking at us here in the US, not in a very kind way right now, because we are giving incentives, lottery prizes, and literally offering money for people to get, vaccinated, while the rest of the world is begging for vaccines. So we are looking spoiled here. We better get the vaccines out of here that are not being used, and literally give them away, because it is in our best interest. It's almost like in health insurance that we are paying. We are paying health insurance, is a health insurance policy here. by minimizing the number of people getting infected, we are, helping all of us, including ourselves here.
Yeah. So Jack asks, what can you tell us about the very new Chinese variant?
Too new, so we don't know much about. So this happens to every variant, right? A new variant comes in, people get really concerned, like with the original Delta variant. It was called the double mutant, at the beginning, remember? Even though it's not technically a double mutant because it has multiple mutations, that type of thing, but it acquired two important key mutations. That's why it was called double mutant. We didn't know much about it until we had that gigantic, quick, explosive surge in cases in India. So I think it's too early to tell anything about the Chinese variant yet. We really need to get those variant sequence, understand, transmission rates, that type of thing. So again, it just calls, just the point that I was making before. Let's just try to get as many people vaccinated as quickly as possible. This is, again, the best way that I can put this is health insurance. For everybody in the world, is a price that we must pay. It's going to benefit all of us. We cannot, close our borders and pretend, that everything is going to be fine if you all get vaccinated and we only allow vaccinated people. No, the virus will find a way to get here. Okay. That's what viruses do, so we have to leave that mentality that, we solve the problem here, let's get back to normal, everything is normal. We are going to have to pay a little bit of a price here, a premium, so to speak, to enhance the health insurance to a level that, can protect us not only from what's happening here, but also from what's happening, elsewhere. So I would argue for more money for research, right? Because it's thanks to research that we got vaccines approved in a 1-year time frame. So the preparation, all of the knowledge that we had about SARS coronavirus, the original one from before. Quite honestly, I was one of the critics about the lack of funding for SARS coronavirus research before the pandemic, before even knowing about this new one. I actually was in a meeting with one of the SARS coronavirus experts, one of the best experts in the world. He's here from the US. And I was telling him, we need to do more about this, and he was complaining to me that, really, because SARS came and kind of went away, the original one, people said, let's forget, there's no rush anymore. And the reality is, if it happened once, it's going to happen twice. And you know what? It's going to happen 3 times, and we're going to have more of this. So we better, keep doing research, improving these vaccines even more, understanding variants, understanding how to develop vaccines that are universal, that have the potential to work against a number of existing variants and even upcoming variants, right? So that's where, we should be putting our money on. We are very smart, people in the world are very smart, not only here in the US, everywhere. We can get there, but we need to pay the price. We need to pay the premium, for that additional, insurance.
Got it. Thank you so much. We could keep you another hour at least, I'm sure, with questions, but we're already just over 8 o'clock. And if there is any last question somebody wants to either shout out or put into the chat.
I think Charles just asked a question that I think is a good one for a person who can't be vaccinated, and there are those people, right? So how safe are they with a mask in a now maskless world? So they are not safe. Okay, absolutely not safe. The virus is around. We are all going to be transmitting this virus at very low levels. Okay, people are not going to get sick. You just don't even know that it's lurking around. Okay, so that person is not going to be safe. So that person in congregate settings, with other people, they're going to have to, at a minimum, be wearing a medical grade mask, because a medical grade mask is so much superior than the ones that are free, the freebie ones, the ones that the university provided, this one here, the ones that my mom made lots of masks in Brazil. She sent me a whole bunch of them. Sorry, Mom, they're very beautiful. They're really very beautiful, but I want the medical grade one. But really what I want for those people is an N95, okay? So an N95 mask. And they are designed for single use, because they have to really fit really well. They have to, when you inhale, the air cannot be coming in from the sides here. They were designed to be used, only once, but I have been using this forever, okay, because it's kind of the only one that I have. When I am shopping, I'm in a supermarket, oh, I'm wearing this, and I'm still wearing this. I'm not planning on not wearing this, okay? I don't want to get the virus and transmit it to a person that cannot get vaccinated. So altruistic, yes, okay. Does it make me feel good? Absolutely. Should I do any differently? No. I mean, I know better than that, right? So an N95, which were so hard to find, right? But now, you may be able to find them. And quite honestly, I mean, if I have to, my editorial for the day is, back in January, February, this is what we should have made. Millions of this, okay? Sure. Millions, billions could be reused, that type of thing. It's a piece of filter that's molded. This one has a valve, but the valve actually is not good, okay? Is good only for other purposes. But this is the only one that I had, right? So, but at least protects me, doesn't protect others. It's molded. I mean, I can't think of anything. I mean, we can go to Mars. We have rovers in Mars now, sampling rocks and things like that. And we could not make enough of this for all of us. There's a problem here, okay? So this is the N95, and there are some other ones that are not as good. Some of them are manufactured in Korea and China. Even those are better than this. So there's this, the ones that come from China and Korea, and then the true N95s. These are really the best ones that will give you the best protection, and that's what these people should be wearing when they go outside the household, that type of thing. Okay, to protect themselves.
Generally speaking, we should all be wearing masks all the time when we're out with people.
Yeah, I mean, so I mean, I still feel uneasy, wearing them. I still have this ethical issue of what if I'm not gonna even know that I'm gonna be infected. I'm gonna be, locally replicating the virus and perhaps transmitting it, and I pass it to somebody else. And I'll probably never know that I did it. But again, I know better. I know that that's a possibility. So I can't live with it. And so I'm not gonna abandon my old one, and probably I'll try to buy a new one finally, because I'm just gonna try to protect them. By the way, when I wear this one with the valve, you always should wear the other one, right? Because when you exhale, you wanna have those droplets, caught by the other one. But the best ones are the ones without the valve. So we need more of those, no question. And we made a critical mistake at about February or so, where we were focusing too much on hand washing on surfaces and disinfectants and wipes, and which I always have here, but even before, because I like to clean my desk here where I eat on my desk, hand sanitizer, that type of thing. We focused too much on those, when in reality the virus was coming in through our nose, not through our hands, And that was sadly a mistake that was made. I quite honestly don't understand that mistake. There was a reason for it. It's because we wanted to save these for the medical, first responders, medical personnel. But we should have had a plan B and say, okay, you know what? China built an entire hospital in Wuhan in 7 days. Why can't we build a plant that makes gazillions of these things, per day? Right. So we failed terribly there with the masks, and that also generated this whole hand washing thing. Generated this idea, now that this anti-mask movement that we have now. Because if you realize early on that masks, really made a difference, perhaps we wouldn't have to have the lockdowns and all of that stuff. But we didn't have enough masks, so that was kind of inevitable. And then everything is snowballed into the situation that we are now, where people are fighting because of masks and you name it. But so a sad reality. I hope that we learn our lessons, but I have a feeling that we humans are just too flawed, I guess, to learn our lessons from history.
Sure. Okay.
I have one more question. Yeah. One last question. So,
could you speak of T cells and B cells, long-term memory cells? Also, how to make your immune system stronger? Vitamin D.
Yeah. So, yeah, absolutely. So, very good question, because that's one of the things that we don't know. Are these messenger RNA vaccines and some of these vector-based vaccines, are they really inducing good T cell and B cell memory? Because that means, that you're going to have those antibodies and T cell responses for long periods of time. Sometimes, like when you get the measles vaccine, you don't need to ever get vaccinated again. Okay, you get it and you have lifelong immunity, long-term immunity. Sometimes you do have to get revaccinated. But we know that it lasts for a long, long time, like our tetanus shot. We only need it every 10 years, right? That type of thing. We don't know the answer to that yet. This is being monitored day and night. So, people in those original clinical trials keep getting monitored because they are ahead of us, right? They started earlier. So they keep monitoring them, and we're gonna, have an idea. And when we see that group of 20,000 people for each one of the clinical trials that actually got the vaccine and not the placebo, once we start seeing, some of those things declining, it will be like, okay, time for a booster. Okay, so we will know that eventually. What you can do, yes, vitamin D, but I wouldn't really just take vitamin D. I mean, vitamin D is good in general, particularly for us here in the Northeast because of our lack of exposure to the sun in the winter. But a balanced diet sounds like a great idea. Our immune system, will perform a lot better if you eat healthy, non-processed foods, low calories. Everything that sounds reasonable, yes, our immune system likes that. Very good data showing the immune system underperforms if you are obese, underperforms if you have a high sugar type of diet, processed foods, all of those things. So everything that the doctors and dietitians have been telling you is correct, even for the immune system. So eat your broccoli, all of those things, do it, okay? Try to do, the antioxidants, everything works in your favor. Number one enemy for all of us is really sugars, because, diabetes and again, obesity, which results from that, really has a big effect on our immune system, and it's not a good one. Okay, so not even surprising. I can't claim that that's the reason why people with diabetes and obese people are more susceptible to more severe disease, but probably has something to do with it. Okay, so that's clearly well established now. So any diet and exercise, and by diet I mean good diet. It's there's no other way to escape it. I would try to stay a little bit away from too much supplements. I would prefer to acquire them in a more natural way. But I understand that some of us can. I take vitamin D myself now because I haven't been in the sun for 15 months. So exactly, and I'm not getting any here, from my office from this artificial light here. So I know I better get some vitamin D. So I'm even giving it to my kids because we have been locked down for 15 months. Normally I wouldn't do that. So so absolutely, if it makes good sense, it makes good sense. It's a good idea.
Too true.
All right. I appreciate, everybody's questions. I really enjoyed them. I'll try to follow up myself on that CT question of the value 28 or so, and try to educate myself a little bit more. But I hope that you guys enjoyed it.
Very, very much so. Thank you very, very much for all of the advice, all of the information, and your way of articulating it so that not just librarians, but everybody else can understand it as well. And I very much appreciate the advice for staying well. And I hope everybody can take it and help pass it along, and go get vaccinated if you haven't. Thank you, Dr. Verardi, so much, and wish you health and wellness. And please keep that message going out as far as you can distribute it, because we'll all be better for it. And thank you for all your work in all the studies and the research. It's life-saving, absolutely. So thank you, and good night, everybody.
Okay, thank you, Anne, and thank you, everybody. Great questions. I really hope that you enjoy this as much as I did, because for me, the ability to talk to people is not something that I used to do much before the pandemic. So the pandemic did create this new opportunity for me to actually talk to people, and that's great. I've been learning a lot myself. So thank you, everybody. Thank you, Anne. I'll be glad to come back another time.
Great. Thank you so much. Take care.
Take care. Bye-bye.
Good night.
Good night.